Hi, in this lecture, we are going to look at the anatomy of a nephron. So the nephron is the basic functional unit for the kidneys. In fact, each kidney has about 1.25 million nephrons, which is quite a bit of nephrons. And the nephron's job is basically to filter the blood and form the urine. Now, urine is what leaves the kidneys. As you remember, it'll go into the renal pelvis and out the ureters, whereas the good stuff, as I call it, has to go back to the blood. So the blood comes into the nephron and it's basically sorted. Any kind of metabolic waste is going to be kicked out through the urine, and anything the body needs is going to need to be held onto in the blood. And if we look at a nephron, this one's uncoiled, so you can see the structures a little easily, but notice the parts of the nephron. So we have this thing here called the renal corpuscle. The renal corpuscle has two things inside of it. It has that capillary bed, which was called the glomerulus, and that's that capillary network all twisty and coiled. And the yellow ball around it is called the glomerular capsule. Now, you might see it called Bowman's capsule. That's the older term for it. So it makes sense. So the glomerular capsule is the part of the nephron. It's kind of like a cup, and it surrounds the blood vessels, which are like my fist in this example. So it surrounds it. The whole thing is called the renal corpuscle. The nephron part is called the glomerular capsule. And what happens is we will form what's called filtrate. So if I say filtrate, then that tells you that this is the material inside the nephron, and the filtrate is formed here from the blood, and then it passes through this wiggly thing called the proximal convoluted tubule, or we just say PCT, and then it goes down. This is called the descending loop. And so we have this nephron loop, or basically it's called the loop of Henle. So we have a descending limb, and then it makes a U-turn, and we have an ascending limb, so it comes back up. And then we have the DCT, distal convoluted tubule, and then we have the collecting duct. So in this image, what we've done is we've blown up a piece of the kidney so that this is the cortex, this lighter area, and then the darker area is the medulla. So that's the flow of the filtrate as it passes through, but there's some adaptations in each part that we need to talk about. So the glomerular capsule, or Bowman's capsule, that's where filtration occurs. So here's a close-up of it. Notice when we look at this, you can see the yellow and the red. So just to get you oriented, we have the afferent arteriole coming into the glomerulus, and then the blood leaves to the efferent. This is the glomerular capsule, and there's two layers. There's this outer layer, which is parietal. Notice that simple squamous epithelial tissue. And then the visceral layer is what you see in yellow. It actually attaches to the glomerulus, and you can see a scanning electron microscope view here. You can see the glomerulus in red, and then you can see the visceral layer. And these cells are called podocytes. They kind of look like an octopus. They have all these extensions, and they wrap around the glomerulus. Now, between the arms are these little gaps, and these are called filtration slits. And so we have the Bowman's capsule, or glomerular capsule, has two layers, an outer parietal, and then you have the inner visceral made up of podocytes, which attach to the blood vessels, to the capillary, the glomerulus. And then between the arms are little slits called filtration slits. So this is going to make this membrane pretty permeable for filtration to occur. Now, once the filtrate is formed, right, it's going to leave the blood and enter the PCT. The proximal convoluted tubule was this kind of twisty part right at the very beginning, right off of the glomerular capsule. And as the filtrate flows through here, a lot of reabsorption is going to happen. So when you see reabsorption, what reabsorption means is that you're moving materials from the filtrate back into the blood. Because you'll see that filtration is not very selective. Anything small goes. And there'll be stuff in our filtrate that we need back. So reabsorption allows it to leave the filtrate and go back to the blood. Because we're doing so much reabsorption, these are simple cuboidal epithelial cells, and they have microvilli. Remember, microvilli are going to increase the surface area, which will really help with the reabsorption. Now, from the PCT, the filtrate is going to flow through the nephron loop, or loop of Henle. It's going to go down the descending limb, make a hairpin turn, and come up the ascending limb. So when we look at this nephron loop, there's different permeabilities to that. The descending limb is simple squamous epithelial, and it's permeable to water, but impermeable to salt. So what that means is that water can leave the filtrate, but not salt. And the salts we're looking at are NaCl, sodium chloride. If we look at the ascending limb, on the other hand, it's called the thick segment. That one's permeable to salts, but impermeable to water. Now, this will be important in a couple lectures, but the salts are able to leave, but not the water. So if we look at the nephron, the one you have in your notes, notice how it's all kind of like twisty here. So what we see is this first part, I'll put a 1 on. This is the glomerular capsule. The whole thing is the renal corpuscle if you include the blood. But if we're just doing nephron, we have the capsule, and then the filtrate goes into this squiggly thing here, which is your proximal convoluted tubule. From there, the filtrate flows down the descending limb. So we'll put a 3 here. And then it makes that loop and it comes up the ascending limb. So I'll put a 4 there. Now, how can you tell? Well, the descending limb is the thin segment and the ascending is always the thick segment. And then it wraps over here. See all this twisty stuff? This is all the DCT. So I'll put a 5 right there, right there, right there. Then it leaves the DCT and it goes out the collecting duct. And that's going to drain into the renal pelvis. Notice when the DCT wraps around, it actually comes in contact with the afferent arteriole. It actually touches, and we'll be looking at that next. So if we return to our rolled out view, we have our DCT. Now our DCT is still simple cuboidal, but it doesn't have microvilli, very few of microvilli. So you can see that there's like a little bit, and it's best suited for secretion. So secretion is when we move materials from the blood back into the filtrate. So we have materials moving back and forth between the blood and the nephron. And if you remember this whole network of capillaries, that was called the peritubular capillaries. And you can see how they're right next to the nephron, to all those tubes. So you can easily move materials back and forth between the nephron and the blood. And that's what's going to happen for forming urine. Now there's 2 types of nephrons. Most of them are called cortical nephrons. And if you look at a cortical nephron, it's cortical because most of it's out in the renal cortex. So most are this type, fairly short loop of Henle, typical nephron that we looked at when we look at anatomy. If we look at juxtamedullary, this is this one, notice that the loop of Henle, or nephron loop, you can call it either one, is really, really big, and it goes all the way into the medulla, so it's slightly different. And it has an extra blood vessel loop called the vasa recta. So we'll look at those later when we look at how we concentrate our urine. Most nephrons are going to be this cortical type, and that's what we looked at, and you had this picture in your notes. This was a cortical nephron. And if we look at the kidney model in lab, notice that you can see that there's a nephron up here, and then there's a nephron over here. So I'll do a 1 and a 2. So this is a cortical nephron, and this is the juxtamedullary. See how deep that loop of Henle goes? So you can see that in that image as well. Now, I mentioned that the DCT actually wraps around and comes in contact with the afferent arteriole. So this piece right here where the blood vessels touch the DCT is called the juxtaglomerular. Juxta means close. So it's next to the glomerulus. So the JG apparatus, or just JGA. So this whole structure is really important at monitoring blood pressure and also filtration in the kidney. And it's just where the DCT meets and touches the afferent arteriole. There are 3 types of cells here that I want you to know. The first ones are just called mesangial cells, and they're like these pinkish cells in here. All they do is support. They don't have a physiology role. They're more of a supporting role, and they help support the afferent arteriole as well as the DCT. And then you have what are called the JG cells, and the JG cells are the ones that are in the afferent arteriole primarily. And the macula densa cells, they're shown in green here. These are the cells inside the DCT. These cells are going to have a physiology function that we'll be exploring more in a later lecture. At this point, I just want you to know that these JG cells in the afferent arteriole mainly are going to have the renin. You call it renin or renin. So you knew that renin was an enzyme and it came from the kidney. Now you know it comes from the JG cells of the afferent arteriole. And those cells basically work like mechanoreceptors. Mechanoreceptors monitor the stretch, and that gives an idea about the blood pressure. When we have filtration occurring in here, filtration is pressure driven. So it'll be very, very important to have, basically control the pressure coming in. The pressure exerted by the blood was called the hydrostatic pressure, and that was directly proportional to the blood pressure. So we will want a mechanism to keep the blood pressure constant here, regardless of what's going on in the rest of your body. And that's what these JG cells and the renin or renin are going to do. The other component here are the macula densa cells, and they are in the DCT, and they're chemoreceptors or osmoreceptors. So they're monitoring the filtrate osmolarity. Osmolarity refers to, think of it as being how salty it is. It refers to the number of solutes. So if the osmolarity goes up, it's more concentrated. And if the osmolarity goes down, it's more dilute. So it's really important at checking the filtrate and making sure the filtrate isn't too salty or too watery, because that filtrate is what's going to be eliminated as urine. And we'll be investigating the function of those cells and how they control filtration in our next lecture. So this is the end of the nephron anatomy lecture.